Okay.
Wait. No. Glycolysis 2.
Yeah. It's a galactose metabolism. Okay. First of all, metabolism of fructose in the liver. The question on the exam is exactly like this. Write the metabolism of the fructose on the liver. You should explain that the fructose by the fructokinase produce first fructose 1-phosphate, then by the aldolase produce glyceraldehyde and dihydroxyacetone phosphate. DHAP means dihydroxyacetone phosphate. Then the glyceraldehyde by the tyrose kinase add 1 phosphate and will be GAP. It means glyceraldehyde phosphate, and it's enter to glycolysis. Important. What is the galactose metabolism? Exactly like an exam question. No. First of all, by the galactokinase. By what? Galactokinase. It will be galactose 1-phosphate, like a fructose 1-phosphate, but it was fructokinase. This is a galactokinase. Okay. And you see, it's forget here to write the magnesium, because magnesium is always everywhere by the ATP also.
Did we went through enzymes? Basic of enzyme kinetics and
Is it there also?
regulation of enzyme activity.
Yeah, we don't go there, and that's very important. Now, UDP galactose 1-phosphate uridyl transferase. UDP glucose galactose. You see double dot galactose 1-phosphate uridyl transferase. What does it mean? It means we once produced. We want to add to consume UDP glucose. Come here, Lisa.
What is TCA?
Tricyclic acid cycle. It's here. UDP glucose and use galactose 1-phosphate, and we want to transfer uridyl by the uridyl transferase. What's happened? Galactose 1-phosphate will be UDP galactose. That's very, very difficult and important. Then the UDP galactose by the epimerase. What is the epimer of galactose? Glucose. And say these two names are similar. Which means this is an intermediate here. UDP4 epimerase, UDP4. Why 4? Because glucose and galactose are epimer at carbon number 4. 4. 2 is monos, okay? Now, first of all, it's consume 1 NAD. Produce lactone here, not lactone, that's a ketone here. Again produce 1 more NADH. It means you can omit this part, but you should write here 2 NADH produce, and that's a UDP glucose.
Can you write where you said the 2 and 4 and 6?
Glucose and galactose GLA are epimer at C4. Okay, and glucose and monos are epimer at C2.
Okay, on 6.
What?
What was on C6?
There is no epimer.
Okay. No, okay.
Epimer means change in just in 1 carbon. Galactose, galactokinase, then galactose 1-phosphate, glucose 1-phosphate, UDP is produced here. What should you remember? Just these two enzymes is important. This enzyme and this enzyme. Galactose metabolism. That's exact exam question. Now, the next page. Galactosemia, it means a high amount of galactose. The galactose by the aldose reductase produces a lot of galactylol, is a galactose sugar or sugar alcohol, and it's caused such enzyme deficiency. Just read this information to know it, okay? As the information.
Okay. We have to know the reaction?
I never saw that. Maybe coming. Do you remember glucose and fructose produce sorbitol and mannitol? It's also that. Galactose produce galactylol. This is just a summarize of the reaction. And is written essentially irreversible is 1, 3, 7 and 10. Okay, but it's just here, and you should put here irreversible also.
But when it's always reversible, why is it like why is it always wrong?
You see, no, somewhere is reversible, somewhere is not, but this is irreversible, I'm sure. when you go to the lecture, it's the teacher tell you that. Now, hexokinase enzyme. Hexokinase, we have 4 type of enzyme, hexokinase. Hexokinase 1, 2, 3, and 4. Hexokinase 2, 1, and 3, dominant hexokinase isoenzyme in muscle. They found in muscle. They have a high affinity for glucose. They are allosterically inhibited by glucose 6-phosphate. What does it mean? What is the job of hexokinase? Add phosphor to the carbon number 6 of the glucose and produce glucose 6-phosphate.
Yeah.
You see, here is glucose, here is glucose 6-phosphate. The job is done. The job of this enzyme is done. Now I have a high amount of glucose 6-phosphate. Come back and said, enzyme, that's enough. Don't produce me. It's written allosterically inhibited by glucose 6-phosphate. Just remember this sentence. Always on the exam, if they ask you, who inhibit this enzyme? Who stimulate this enzyme? Always the product inhibit and said that's enough. Please don't produce me anymore. And always the reagent said, please stimulate, please be active and change me to the product. Understand? It means that if you know this rule, that is very easy to memorize all of them. Always.
The product is for inhibition and reagent is for stimulation.
Yeah, inhibitory allosterically is product and always stimulatory allosterically is reagent. And not just in one reaction. For example, if A produce B, B produce C, C produce D, this enzyme is stimulated by A, B, C. And this enzyme is inhibited by D, E, F, G. All of the chain, all of the product. Other product also can come and inhibit that. Okay, that's enough. Please don't do anything more. Now, come here. This is a fructose 6-phosphate should produce fructose 1 and 6-bisphosphate. What is the job of that? This is in the direction of glycolysis or inversion of glycolysis. When the fructose 6 will be fructose 1 and 6, this is a glycolysis.
Yeah,
It's go down. What is the end of the glycolysis on the citric acid cycle? Produce lot of ATP.
Yeah.
If the ATP is high, this should happen? No, it's inhibited because the ATP is a final product. But if the substrate of ATP, AMP and ADP is high, activate it. Understand?
To produce ATP.
Yes, to produce more, to go down and produce more ATP. If the citrate is high, where is the citrate here? If the citrate is high, said please glycolysis, stop. Please don't do anymore. I'm a final product. But if the fructose 2 and 6-bisphosphate is high, stimulate it. Just forget this one, that's not so much important. But know the other is important. Okay? You should know all of them. Inhibitory, stimulatory.
They can ask like if citrate is high, what happens?
You see, first of all, they ask the name of this enzyme. It was phosphofructokinase 1, PFK1, the name of this enzyme PFK1, phosphofructokinase 1. They ask it, could you please write the reaction of phosphofructokinase 1? The second part. Could you please mention who allosterically inhibit this enzyme? Third part. Could you please mention who's allosterically stimulate this part? Together 10 points.
What is allosteric?
Allosterically. I told you last week. Do you remember GABA?
Yeah.
GABA is in the brain. Diazepam stimulated, phenobarbital stimulated, chloride stimulated by different location. If, for example, you imagine this is an enzyme, from this part is stimulated to be closed.
It can be in both directions.
Yes. In other one.
Yeah.
But in this one, I can inhibit and said, no, please don't come in. It means it has a place to stimulate, it has a place to inhibit. This is an enzyme, has an allosteric regulation. Happens on that. But such an enzyme don't have allosteric. Where? In the laboratory. In our body, all of them has. Okay? Now, come here. Hexokinase 4, the other name of hexokinase 4 is glucokinase. Is predominant in the liver, not in the muscle. That's first difference. Okay? Lower affinity for glucose. What does it mean? It has a high affinity at 0.1 millimolar. This is 10 millimolar. What does it mean?
So it needs more glucose to react.
See, I eat sugar. Sugar is always high in my liver, yes? When the sugar glucose comes to the liver, it should be stored as a glycogen, storage energy. But the glucose comes to your muscle.
Yeah.
The muscle wants to do a sport. There is no energy. It takes a lot of time that the glucose is break down from glycogen on the liver, comes to the muscle, transported into the fiber, actin, myosin on the mitochondria to produce ATP. It's a high time. In this case, the God put hexokinase 4 on the liver and hexokinase 1, 2, 3 on the muscle. Actually, if you want to thinking better, hexokinase 1, 2, 3 is in the muscle, and they said if we have a 0.1 millimole glucose, that's enough for us to produce 32 ATP from 1 mole glucose.
32 ATP.
32 ATP.
64 ATP.
Understand me?
96 ATP.
But on the liver said, I see a lot of money, I see a lot of glucose. I don't care if 0.1 millimolar comes. If you comes to be 10 millimolar, okay, I start the car and I do something for that to produce glycogen from that. Because I see a lot of them. I don't need a little amount. Do you understand? Mm-hmm. The muscle always doesn't have so much glucose and energy. 0.1 millimolar is enough. It means high affinity for a little bit money and energy. This is hexokinase 1, 2, 3. But hexokinase 4 is in the liver. 4, highest number, higher affinity to glucose. Half saturation is much more. I hope you understand.
So you,
because this is 200% important to compare them.
In muscle, they're like, you need little glucose in order to produce many ATPs.
So we need a lot of energy.
Yeah.
But there is no much more glucose.
Yeah.
In this case, the enzyme here are sensitive also to low amount of glucose. They said we need energy. We need money. If you put me 100 fruit, I said thank you so much, madam, because I need it. Okay? But the liver is saturated. You give him 10,000, he says no, I don't want, give it back. I should put my hand just for 10,000? Minimum 1 million for him.
Okay.
Maybe I said you thank you. Understand me?
Yeah. Wait, I just need to see one thing.
No, it doesn't finish.
It is, yeah.
It doesn't finish. No, it's here. It's not inhibited by glucose 6-phosphate. What is the job? The glucose 6-phosphate was the
GCP.
was the product, and said, okay, that's enough. I do it on the muscle. Please don't use anymore. But on the liver, it doesn't stop. Why? It's inhibited. It doesn't not
Because
inhibited.
we can always produce glycogen and it can be stored
You see,
there.
if this is 10,000 is not enough, 1 million glucose, 1 million foreign glucose should come on the liver. If 1 mole wants to produce glucose 6-phosphate, it's inhibited. What the 999 wants to do? In this case, the glucose 6-phosphate shouldn't inhibit because there is a lot of glucose and all of them should be stored like a glycogen or should be produce a lot of ATP for the body. Understand? This case shouldn't be inhibited. It's just inhibited by the regulatory protein. For example, you later learn about of insulin, glucagon, cortisol. Just like this. No. Regulation of pyruvate kinase. This is a pyruvate kinase. That's the enzyme, okay? This is active when there is no phosphor on that. There is inactive if it has a tail. This is a phosphor tail. It's very heavy. It cannot move anymore. Who add this phosphate? ATP. By the help of the pyruvate kinase activation. Who stimulate this job? Glucagon. Why? What is the job of glucagon?
Store.
Break the glycogen, produce more glucose.
Yeah.
Glucagon break glycogen and produce more and more and more glucose. What is the job of glycolysis?
Produce ATP.
It means usage of glucose. Usage of glucose. But my body needs glucose. The glucagon wants to produce glucose. The glycolysis wants to use the glucose, or vice versa. The glucagon doesn't like glycolysis. It means all of the enzyme could be inhibited by glucagon. And insulin is opposite. It stimulates all of them. Please use glucose. Okay, insulin like glycolysis, glucagon doesn't like. Insulin like glycolysis. It like means stimulate. This symbol, glycolysis, and glucagon hate it. Glucagon hate glycolysis. Because of that, try to inactive the pyruvate kinase. Again, ATP, acetyl-CoA, long chain fatty acid, these are the final product. Inhibit this enzyme and said, okay, that's enough. We are a lot. Why do you want to produce us more? But the reagent stimulate and said, please be active. I'm a lot here. I'm waiting on the Q. How do you pronounce it? Q. I'm waiting on the Q. Please be active more and use me. Pyruvate alanine, if there is a lot of alanine, said that's enough. Please don't me produce again, because it's in the exam. Please, regulation of pyruvate kinase explain. That's important.
Explain. So we just draw this structure.
Is it 200?
No, all of the regulation are 50%. No, glycolysis inhibition. Always you see the product has a inhibition, ATP, AMP inhibition, but it doesn't write here because they don't have an allosteric regulation. Just these pyruvate kinase, phosphofructokinase, hexokinase, has an allosteric regulation. Maybe a stimulatory, maybe inhibitory. Because it was before here, before.
Oh, how important is that?
You see, actually, this part is important. Glycolysis inhibition happens by that. Now, glycolysis activation happens by the AMP. That's very difficult here. This is the ATP divided by AMP. The name of that is ATP-AMP ratio is low. What?
Is low.
ATP-AMP ratio is low. Is low means the denominator is high. It means the fraction will be down. If the ATP-AMP ratio is high, it means ATP is high. Now, if the AMP is high, stimulate and said, please produce more and more ATP. There is no lot of ATP. Understand?
If this is high, AMP is high.
Yeah, because it's the substrate.
This is the ATP to AMP. Good? If this is high, it means ATP is high. It means inhibit glycolysis. ATP to AMP is low. It means ATP is low. It means stimulate glycolysis. Understand it?
Yeah.
No. What's this? If we use glucose, this is glycolysis. If we back from pyruvate to produce glucose, this is the gluconeogenesis. Genesis means generation. Neo means new. Newly glucose generate. Understand? Now, all of them come here, they back. If the reaction is reversible, there is no any problem. It's come here, it's back. It's come here, it's back. What reaction number 1, 3, 7, and 10 is irreversible. The 7 can manage it. 7 can manage it, okay? But 1, 3, and 10 is vice versa. This is very difficult, confusing. What's happened? Phosphoenol pyruvate by pyruvate kinase produce pyruvate. When pyruvate wants to back, first of all, by the pyruvate carboxylase, carboxylation happens on pyruvate. Pyruvate, how many carbon?
3.
3. If I add 1 carboxyl to the pyruvate, it will be 4 carbon and oxaloacetate produced. Do you know what is the structure of oxaloacetate? COO minus, COO, C COO minus. Is the oxalic acid and acetate. Then phosphoenol pyruvate carboxykinase produce phosphoenol pyruvate. And you know that structure also. It means we have two enzyme name. One of them use ATP, one of other two GTP. It means 4 molecule energy use. How many?
4.
Okay, let's go up. Fructose 1 and 6 bisphosphate wants to back. Here is a fructokinase.